background, okay, yeah. So being from civil engineering background, I cannot forget cement and clay. So we work with polymer coating on cement on concrete surface. And particularly, we look at the interface of the cement and this concrete, and also cement and polymer, and also polymer and clay. That's another one. And the third one, which is where we look into the fundamental aspects of the polymer water interactions. This we do primarily with the molecular dynamics and simulations. We look into different mechanisms involved. And there is one small discussion that I will have today in this presentation on this. And recently we started exploring the different mechanical aspects of molecular crystals with Professor Pradeep's lab. So these are the four major domains of our work. Now, as I said, I will be talking mainly on this first one. So keeping the interest of this particular seminar series in mind, what I will do is I will not go into the details of any of our work or the results or anything, but I will be primarily bringing a couple of problems on the table, and whatever necessary background is required to define this problem, I will discuss about that in very brief. And the part B, maybe last 5 minutes or so, we'll talk about some expertise that we can extend and see if we can help you in some of your works through using our nano indentation or technique or nano indenter. So that's how this talk is designed. So now solvent triggered actuation, soft actuators, and sensors. Like polymers, they respond to different types of triggers, like very commonly known are magnetic field, electric field, and things like that. However, recently we see that there are different other triggers coming into picture, particularly when the matrix is polymer or hydrogel. And one such is solvent. Besides that, we also work in collaboration with other colleagues here with the light triggered and vapor triggered. But in our lab, we primarily focus on this solvent triggered, which I will be going into a little bit into the details. Now, what actually happens? Well, when you actually put a thin film, polymer thin film on a certain solvent, it undergoes a folding like this or outer plane bending. And then you can imagine or you can extrapolate this phenomena to different applications. So that's what happens. And here it is, the substrate is Chitosan, which is a biopolymer. The functional group is given here. And in this particular example, this thickness is about 50 to 80 micron thickness, and the length we are talking about 7 millimeter and the width 3.5 millimeter. And the typical folding unfolding time that we are talking about in this particular example is about 20 to 30 seconds. But all these parameters can be actually tuned by tuning the material properties. Now, as far as this actuation is concerned, normally the 2 or 3 very common parameters that we characterize that we actually measure to characterize this film folding are the total time of folding, how much time it takes, the actuation speed, that's very important, at what speed it is actuating, and the curvature, because when it comes to specific applications, this curvature becomes a very important parameter. And of course, there are certain other secondary parameters also there, which depends on your applications can become primary. For example, the gripping force, how much force that it apply when it is trying to grip something. That's another important thing. So, but typically these are the 3 fundamental parameters which characterizes this actuations or this folding. Now, let me slightly go into the details of what exactly is happening when we are actually when we're seeing this folding. Now, when we imagine this film to be kept, on a solvent. Now, and then you assume a thickness of, from somewhere from the middle. Now, when the bottom, and this is, let's say, the bottom, and this is at the top. Let me take my pointer. Yeah, so this is the bottom and this is the top, and the solvent is actually going from the bottom to the top. So when the solvent molecules, when the polymer comes at the bottom, comes at the proximity of the solvent molecules, the solvent molecule enters through this and moves towards the top through the diffusion. Now, the concentration of the solvent molecules at the bottommost layer is much higher compared to the top at the beginning, and then slowly the concentration actually, it gets uniform as time goes on, right? So there is a concentration gradient, as we can see from this schematic that exists, And to accommodate this additional amount of concentrations, or you can say the amount, the number of what solvent molecules, the only option for the film is to actually bend. Very similar to thermocouple, but it is slightly different in other sense. So number one is, the first thing is that there has to exist a concentration gradient for the film to fold, number one. Now, the second thing is that as these water molecules or the solvent molecules are going into it, now what happens is it changes the polymer configurations and it changes the chain conformations. Now, because of that change, it actually is affecting the mechanical property of the polymer. Now, this change in mechanical property in turn is actually affecting the diffusion characteristics of the solvent into that domain of polymer or into the matrix. Now, thus we can see that it is actually a diffusion deformation coupled phenomena. So diffusion is affecting the mechanical property, and the mechanical property in turn is affecting the diffusion characteristics. So that's, it's a diffusion coupled deformation phenomena that is happening here when it is actually it is trying to fold. So with this, we will go to the thing is that, okay, what is happening when it is unfolding, or why is it unfolding? So this part is not very clear, but we have a hypothesis, we have predicted a hypothesis, and we have and then we have communicated this to soft matter. So what is happening is we are telling there is a competitive mechanism going on. What is the competition between? Competition between the concentration gradient, as you see here during the folding, which is actually trying to give the generate the moment and causes the folding. On the other hand, since these polymer chains or the molecules are actually moving away from the equilibrium and they are either in compression or in tension, it is losing this entropy. So the molecule, so the polymer is actually trying to go back to its equilibrium. Now there is a competition between these two. Now during the folding, when the concentration gradient is very strong, the folding wins. However, when this concentration gradient is actually becoming almost uniform or gradually become uniform, then the second phenomena where the polymer is trying to move, go back and relax and attain its equilibrium, that mechanism is actually winning, which leads to unfolding. So now we said, okay, how do we actually confirm this? Then we said that, well, if we can cut down this ability of the polymer to actually relax or go back to its equilibrium, then it should not unfold, right? That's what we can say. So we said, okay, let's do that. So what we have done is, in the solvent, we have introduced resorcinol, which is, as you know, there's a 2OH or additional OH to phenol. And then this, immediately what we have seen is at a certain, a specific concentration. Yeah. So at a critical concentrations, what we have noticed is that you see that it folds, but it doesn't unfold. So it leads to an irreversible folding. And what we are telling is that, well, that could be because this resorcinol or this organic alcohol is actually sitting between the two polymer chains and not allowing the polymer chain to actually undergo to relax as it would have been, it would have gone in the absence of it. Okay. And to probe further into it, we have done some simple SMD steered molecular dynamics study, and to understand what exactly going on. And we have seen that the dihedral angle is the rotational, the range of this dihedral angle is significantly cut down in the presence of the resorcinol, which is not allowing, therefore, the polymer to go and relax. That's about the irreversible irreversibility. Now, these are some of the some of these conformations or some of the shapes that you can actually get by making it irreversible. Now, just want to share a little bit about this interface engineering of a bilayer system. So we from single layer we move to bilayer because there are certain applications where it demands bilayer, particularly, for example, if you are talking about underwater gripping or folding. Then then if you have the solvent coming from the both sides, then you will not be able to develop that concentration gradient necessary for folding, and thus it will not fold. So you have to put a coating or a hydrophobic layer at the one side to actually cause the folding and when it is immersed in water. And the other thing is that multi-triggered folding. For example, one trigger is not always efficient and good, particularly for a specific application. So maybe you will use one trigger for folding and the other trigger for unfolding. And there, usually it is found that one particular polymer is not effective as far as response is concerned to both the triggers. So that's also where you will probably need a bilayer. Now, when the bilayer comes or multi-layer comes, interface becomes a very, very important factor. So what we said is, can we actually engineer this interface and see if that through that you can obtain the different actuations characteristics. So what we have done is we have done a very simple experiment, and we have actually developed this interface and like 3-4 different types of interface. One is very strong, and then weaker, and there is no interface. So we have basically a different strength, the interfaces of different strength, gradually reducing strength. And the way we have done is, we have a PMMA layer at the top, which is hydrophobic, and then Chitosan is hydrophilic. We have taken help of silane, and then we have varied the concentration of this silane to actually reach to different strength of this interface. So that's how we have obtained a different interfacial strength. And I'll just give a summary of the results that we have obtained. One is, let me play this. One is with one is a strong interface, and the other one is a weak interface. And what we have noticed, I will not get into the details, but what we have noticed is that in case of a strong interface, the folding is permanent or irreversible. And there is a reason behind that. We'll not get into that, particularly because it leads to a permanent concentration gradient. And that's why it doesn't allow it to unfold. Whereas in case of a no interface, when you do not have interface, it's a reversible folding. And the actuation speed, there's a significant difference which you will not be able to see here because it is not, I think, in a properly showing. But the higher actuation speed in case of a strong interface, whereas in case of a no interface, there is a lower actuation speed. And also the gripping force that with it grips is for the strong interface, it is larger than the weak interface. So the summary is, well, we can actually engineer the interface as well to obtain a different actuation characteristics. That's what we actually, and this manuscript is under review. We demonstrated. So now let's come to the problem. So this is, as I said, we'll have 3 problems. Now let me come to the problem 1. So you have seen that it needs a concentration gradient to actually cause the folding. It's a coupled deformation diffusion problem. And therefore, we can actually play with the matrix property and solvent property, as you have seen we have done by adding a resorcinol to manipulate this actuation behavior or actuation characteristics. So now the question is here, like, how do we obtain different patterns of this, right? The different patterns of this folding. Well, there is a lithographic technique that definitely we can do. For example, one, it is demonstrated here, as you see here, that if we can actually selectively put certain stiffener or certain materials with a different diffusion behavior characteristics, then we can get some sort of a pattern like this. And also, for example, this is done manually, where we manually put some hydrophobic mark at 45 degree, and then you can actually obtain a different, it is a curling behavior. However, it is not very, what to say, a robust way of doing. And also for doing a lithographic technique on hydrogels is a little challenging. So what we are thinking is, if we can come up with some novel materials, and then we think of distributing on the matrix itself, on the, for example, along the across the cross sections, or on this is a top view of the film, and this is a cross section. So we can have a distribution of such materials which leads to a differential rate of actuation and relative stiffness. Then probably we can get a different patterns. So basically, we are trying to see that let us introduce certain materials in a certain pattern, so that it will give rise to a different out of plane bending or out of plane folding. So that is one problem. So we want to avoid basically lithography technique here and try to basically modify the matrix itself with certain materials or certain nanoparticles, so that we can get a different types of folding. That's one plan that we have, on one problem that we have on the table. So that is one. Now let's move to the second problem. So before that, let me give the background of it. So this is, as you see, a solvent supplied by coated layer underneath. Now, why coating? So you see, if you've seen what I have shown so far, that you need to put these films on certain solvent, or you have to dip inside a water or a solvent bath. Now, if you think of certain applications, it is not always possible to have such a bath around or such a continuous medium. So how do you apply then? How do you use this then? So what we thought is, if can we come up with a coating, whether at the top or at the bottom, doesn't matter, and the coating that we have will have the ability to hold water, and that water will be released because of certain trigger, and this released water in turn will be supplied to chitosan or the polymer film, which will undergo folding. So that was the idea. So we explored, and then we have thought of using PNPAM, which is polyisopropyl acrylamide with this functional group. So what this PNPAM does is, as many of you know, that when it crosses LCST, that which is low critical solution temperature, what it does is it releases the water, significant amount of water, and it shrinks. So we thought that, okay, can we make use of this water that is released by PNPAM actually to cause the folding of the chitosan, right? So that's what we have done. So we have put a coating of PNPAM at the bottom, and then we raise the temperature above its LCST. And typically the LCST for PNPAM is 32 to 37 if you can actually tune it, but that's pretty much the temperature range. And so that's what we have done. And then we have actually seen that you get the folding because of the water supplied by the PNPAM. And also what we have done is we have actually varied the layer. As you see here, the chitosan here. This is the chitosan film, and this is the layer of PNPAM. And then this PNPAM supplies the solvent for the film chitosan to fold. Now, with the increasing L, you can actually vary the curvature, because the source or the amount of water and the length of the PNPAM is different. The curvature obtained is different. And this is our little cute butterfly, which actually starts flapping its wings when the temperature is raised to 35 degree in this particular case. So and the PNPAM layer was only put at the in this part of the body of the this butterfly. So this is just to demonstrate about that the actuation you can do without even coating the entire length of the film. Okay, so similarly, you can actually put up make a coating and then at a certain angle, and then you can get a specific type of folding here. Right. And then if you want to have certain like this type of a folding where you actually have a coiling, you need a coil to coil around around a certain body. Then you basically what you do is you taper it along the length. And this tapering, what it does is actually it gives rise to different stiffness, the mechanical stiffness, E times the modulus times the moment of inertia. And that essentially gives rise to the different local curvature. And as the temperature rises above 35 degree, it undergoes a coiling like this, and you can actually get this type of coiling behavior. All right. So I will come to the problem now. So this is fine. We're happy that okay, this our concept or hypothesis is working nice and fine. But the problem here is that if you want to use the same system multiple times, then the water that is going to PNPAM, sorry, the water that is going from this PNPAM layer to the chitosan, the same water layer needs to come back to the PNPAM so that during the next cycle of application, it can be used again. So the water or the solvent, whatever, you can put any other solvent as well. So the solvent or the water that is the PNPAM is supplying, that needs to be cycled between this chitosan and the PNPAM. That's what it should be. It has to be, so that the system is more effective. Now, what we have seen and done is we have played a little bit with some different hydrophobicity and hydrophilicity and introduced inside the chitosan. We have to some extent got the water, could get the water back to PNPAM, but it is not coming back, 100%, not even closer to 50%, I would say. So as a result, what is happening is after 1 or 2 cycles, we see that this cannot be used anymore. Now, this is going to be a very interesting problem if we can actually chemically modify Chitosan and or PNPAM so that when your trigger temperature is gone, it supplies the water and the folding. Now, when it is going back to the normal temperature, the 100% of the water that was in Chitosan comes back and hydrate the PNPAM. If that can be done, then this is actually going to be a very useful system, particularly for sensing purposes. So this is the problem.
Use 21 minutes. Huh?
Yes. So this is the problem number 2 that we have. Okay. So I'll talk to briefly about another work that we do as far as polymer physics is concerned. Now there are different types of water in hydrogel or polymer, particularly if you look at wood, food, or different foods or different types of foods. Yeah. Now, this hydrogel or the polymer, they have different types of water. So when I say different types of water, it means their waters are either free water, loosely bound water, or tightly bound water. Now, one of the configuration that the water can be is like this, where it is at the proximity of the substrate, it is very tightly bound, then it is loosely bound, weakly bound, and then it is a free water. But there are a lot of hydrogels and polymers where it is not like that, but it is actually randomly distributed like this. So some free water, some bound water, some tightly bound water, things like that. And what we have seen is that with the temperature, if you do the mechanical property if you find the mechanical property, we have seen that there are 3 different regimes, which basically tells that the 3 different types of waters are actually are escaping from the system. So that motivates us to model this system, both experimentally investigate as well as build the mechanics-based model. But the problem that we have is, maybe from your experience, you can tell what we need is, and what is very important is, how do we determine the distribution of 3 types of waters in a polymer thin film? When I say distribution, it's not necessarily the quantity, but we're talking about the spatial distributions of these thin films. So that is another thing that if somebody can help us, would be very nice. Okay, so I will take 2 more minutes. So that's all about the 3 problems. Now let me briefly tell you about what we can offer as a part of our expertise. So we have an equipment called Nano Indentor, and what Nano Indentor essentially does is, in a summary, it gives you the mechanical property of your system or of your material. How it does is essentially what it does is you have a probe, and then you push the probe, and then you essentially measure how much resistance the probe is experiencing to push it to a certain depth. So as you see here, the y-axis will give you the load, and the x-axis is give the displacement, and the load displacement characteristics will tell you that about the nature of this material. Okay, and the two most important characteristics that normally you measure to actually find out the behavior of this material is the contact area that it has made and the contact depth, like how this contact depth, where it is losing the contact. These are the 2 fundamental parameters that we measure to essentially tell about the mechanical property of the material. Now, this is important to give you an idea about the sample size. Now, the sample size can be anything from few microns to centimeter. Now, this T, this total T that you are seeing, the sample thickness, that actually depends on the H, how much H you want to go. And here we can do a very thin film from nanometers to any bulk material. Tip diameter typically go from 50 nanometer to 500 nanometer, and the width can be, the B can be anything depending on the diameter of your tip. Now I'm telling all these things so that you can connect these to your material. Okay, so what exactly it happens is, so you indent it, and the materials underneath that indenter, because of the load, it undergoes a changes in its configuration. Now, some of these changes are actually permanent, which leads to the plastic deformation, but some are actually elastic deformation. And depending on the material, these regions are going to be different. Okay. Now, these are the load displacement characteristics that you will obtain for different samples, and this actually gives us a lot of idea about the different characteristics of material. And there are several different reasons why mechanical properties of crystals. For example, I'm talking about crystals, it can be thin films. Why it is useful, and you need to find out. There can be a stress-induced transformation. This is a lot of interest to people these days. You would like to find the fracture toughness of the crystals that you have actually synthesized in your lab. And also you find the bending rigidity and flexibility of the certain plastic crystals. That's where also you can apply nano indentation. And for example, you are not able to know whether it is isotropic or anisotropic. So if you just use a schematic representation, so if you have this and then you do a mechanical probing, the anisotropic can be picked up if it is properly done. And then you can tell, okay, this plane is showing this type of anisotropy as opposed to the other plane, which is not like this. You can also talk about about the slip planes, how the slip planes are oriented, what is the force necessary to cause for the slip to actually shear. So these are some of the things that you can actually characterize from using nano indenter and by applying on your crystals. Okay, these are some examples from this. And even you can pick up actually the difference between the hydrated crystals and whether it is dissolving and whether it is picking up water, and by applying a nano indentation. Of course, you have to do it very carefully, find the right load regime, and sometime you may have to do in the dynamic system instead of static. All right, so polymorphs can be picked up. This is something that people use a lot these days, polymorphs and domain coexistence of your crystals. And nano indentation is extremely useful, and there are a couple of published results, and we can also do with our nano indenter. All right, like you are probing in this way, you can also do in the scratching and find out some properties of your crystals. So that's it. I think I would request you to see and think about how this indentation can be actually applied for your system. Recently, we have with Professor Pradeep, we have published these two work, and a couple of works are ongoing, and these are on the mechanical. Investigations of organic crystals or cluster molecules. So that's it. That's the summary. So we have these three problems, which is where we need your collaborations or your expertise and suggestions. And as far as our expertise is concerned, we can extend expertise of our nano indentation to you to characterize your crystals, because I know a lot of people in physics and chemistry, they do synthesize different types of crystals and thin films. Okay, so with that, thank you very much for your time.
Thank you. You have consumed about 28 minutes, so we have just a short, time available for questions. So I will just take one. I will pose one question and ask others to. So one is, all of these interactions are extremely solvent dependent, and you talked about water, and obviously there can be a number of other solvents. So there are many things that come into one's attention, like sensing, right? Solvents sensing is one thing. Vapors is solvent need not be liquids, it can be vapors, and these are getting into thermodynamics. Essentially, this is to do with when you say hydration that is happening, there is certain kind of thermodynamic advantage, and then you are reforming that structure. Energy has to be supplied back into the system. So there is some substantial thermodynamics and associated kinetics with these processes. So do you have insights on these, or are you pursuing sensing, energetics, kinetics of these processes as well?
I mean, with this particular work, we are more going towards the application side. We have not explored that area. So, for example, our next objective is to bring in multiple triggers, like a couple of different triggers, and integrate them together for a specific applications. So, so I would say right now in our lab, we are going more towards the applications and integrating multiple triggers rather than going into that aspect. But Professor Dilip is actually looking into more into the fundamental aspects of this polymer, where for this vapor trigger and this solvent triggers.
Thank you. Are there other questions?
Yeah, hello. This is Abhijit. Hi, Piyush.
Hi, Abhijit.
Yeah, I just wanted to understand, maybe I'll take it offline, the answer may be long. Why is it important to get directly the spatial distribution of water type? Right? As you said, it's maybe relatively, doable to characterize the amount in a film.
Yeah.
But you said you specifically wanted to experimentally get the spatial distribution.
Yeah, I will just briefly tell you. It is because we are developing the model where we have a different types of water, continuum scale model. Now, we are starting with something with randomly distributed, But then what we understand is the random distribution is perhaps okay when you are talking about the bulk polymer. But when you go to a thin film, where the substrate effect comes into picture, as we understand from the literature, that's not a good assumption to make that the different types of waters are going to be randomly distributed. So in that case, we might need a very specific spatial distribution of these different types of water to model it correctly. That's to just to briefly answer you.
Vishal, you have a question. Vishal, you have a question. Go ahead. Okay, can't hear you, Vishal. Maybe there is a network connection issue. Okay, you can come later on. We are just about 31.
Pradeep, sorry, actually, my some problem is there, so my hand raise option is not there.
Okay,
so
it doesn't
I
matter.
don't know.
So you carry on.
Can I ask question?
Go ahead, please.
Okay, thank you. Peter's very nice talk. So I have two questions. One is you talked about this folding and unfolding. It is due to the it is purely due to the structural, or it is also there is some kind of charging effect is also there. I mean, some electrostatic effect may also be there. I mean.
I don't know the second aspect, the charging, because we have never thought in that direction. Like what type of, whether the charging can actually, the charge effect can actually lead to that significant amount of curvature change. I don't know. We have never actually thought that that could actually be one of the reasons. Maybe I will talk to you again in details what exactly you mean and is it really possible or not.
Okay. Okay. And second thing is you somewhere you told that bilayer structure. So this is you told that two materials with hydrophobic and hydrophilic in nature.
Correct.
So how you actually compose those materials?
We have used a silane. So if you use
that would be, I mean,
we should move on. We should move on. Okay. Yeah.
But yeah, we use chlorosilane. I can tell you that how we have done. What is the concentration we have used? And also we have seen that there is no delamination, particularly when you have a certain critical concentration. So,
Vishal, you have a quick thing, otherwise we will move on.
Okay, yeah, sure.
Well, I know we need to close, otherwise what happens last time, we do another half an hour more. So we are trying to reduce that. Vishal, you don't have, I can't hear you. So the next speaker then is Professor Ravindran. So if you can share your screen and then get going, as I said before, 5 minutes, 20 minutes, I will give you a warning.
Good evening, everyone. And it's wonderful thunder outside, and we're all hearing it on our mics as well. It's amazing weather, and really, Professor Pradeep, I thank you for actually organizing this and also for inviting me to this. So what I'm going to do is I'm going to close all my other windows except my slides. So if anyone wants to ask me a question, just speak up, okay? Otherwise, I would be able to see what's happening. Great. I'm going to talk a little bit about the work that my group has been doing on complex graph structures. As some of you might know, that I kind of split my time between working on learning with graph structured data, as well as looking at reinforcement learning. And I thought for a larger audience, perhaps the work that we do on graphs would be more interesting. And I've kind of organized this like a kind of a retrospective, maybe a few slides on some of the selected works that we have done. And I'll be happy to talk to people later about in depth about any of these pieces of work. Right, so graphs are everywhere, right? They're used to model all kinds of phenomenon. So what I mean by a graph here is that, okay, there is a set of entities in a social network that would be people, or in citation networks that would be papers. And then the links between the entities are modeled as edges on the graph, right? So it could be a friendship link, or it could be a citation, right? Or it could be other kinds of phenomenon, transportation, where you have stations connected by railway lines, or biological networks where you have proteins and interactions between them modeled as edges and so on and so forth. So just to tell you that the graph is kind of a universal model that spans multiple disciplines, right? And then I wanted to make sure that people understand what I mean by complex graph structures. I'm going to talk about two different kinds of complex graphs, right? The first are what are called hypergraphs. So I was telling you that in the graph, an edge models a relationship between two entities. But oftentimes we are looking at phenomenon where there are multiple entities involved in an interaction, not just two entities. In such a case, the natural model is a hypergraph. So where you still have your nodes, right, which are the entities, but then instead of an edge connecting two nodes, you'll have an edge that connects multiple nodes. So here you can see that this is a hypergraph denoting students who are in the same class, right? So then so here is one class that has 5 students, you connect them all together. So apart from that kind of a toy example, it also comes up in a lot of biological phenomena. For example, when I'm looking at modeling signaling pathways, so I could actually model them as pairwise interactions using a graph, but typically there are more than 2 proteins that are involved in a specific pathway, in which case I will actually model them as a hypergraph with all these proteins being connected through a single edge. So that's actually a lot of such natural phenomena which lend themselves to hypergraph modeling. And the second kind of complex structure I'm going to talk about is or multi-layer networks, where you have a set of entities, but they are linked by different kinds of relations. So on the left, you see an example of a transportation network. So you have different cities and stations. They could be linked by a train, right? They could be linked by buses, or linked by air, or linked by ferry. And there could also be interchanges. In a particular city, you might be able to move from the airport to a ferry and so on and so forth. And again, since I do a lot of work with the biotech folks, I'm going to give a lot of examples from biology. If I say anything wrong, Adi can correct me. So here again, you have this kind of multi-omics kind of an analysis where you have a DNA layer. So where you have a DNA layer and a protein layer and a metabolite layer. So there are interactions within each of these layers, but there are also interactions that go across layers, right? And oftentimes it makes, it's actually important that you should distinguish these two kinds of interactions. So it is not enough that you can just model it as a graph by collapsing all the edges into a single set of edges. So we'll see as we go along. So there are many different kind of applications. Well, when I'm talking about applications, I'm still in an abstract space, right? I've not gone down to a specific domain yet, but there are many applications that you could define on graphs, right? So on this slide, I only show the different applications that my group has tackled. Okay. And it's range from, at the node level. So at the entity level, you have certain kinds of inferences that you have to make, or it could be at the edge level, right? Or it could be doing some kind of reasoning about the whole graph, right? Or it could be other kinds of properties or dynamics on the graph, like looking at certain evolving processes on the graph and so on and so forth, which kind of more into the dynamical systems defined on this kind of a graph substrate. So these are the 4 different classifications I'm talking about. And in that, I'm going to today talk about these 4 problems: node classification, clustering, ranking, where I'm going to assign some kind of a centrality or importance score to the nodes and rank them according to that. And then finally, I'm going to talk about link prediction, which is to suggest new relations that have not been observed and which are likely to exist, or recommend new relations that should exist. For example, friendship on a LinkedIn graph or anything. So I'm moving on. The first thing I'll talk about is the ranking problem or the node centrality problem. And this was a question that was motivated by looking at a specific question in biology. This is work done with my colleague Manikandan in the computer science department, and our joint PhD student Tarun Kumar. So the project is focused on discovering genes which are responsible for inter-tissue communication. And the way we did this is looking at each tissue in the body as a separate layer. Remember the multi-layer idea. And then there are interactions within the layer so within the tissue, there is a gene interaction network that has going on. But across the tissues also, different genes mediate certain kinds of reactions in the other tissue. So there are these edges that go across tissue. So kind of abstracting away from the biology, we just look at it as a multi-layer network, right? And then we wanted to look at nodes that are important only for this inter-layer communication, not the intra-layer communication. right? So the classical, ranking algorithms for nodes like PageRank and so on and so forth typically do not differentiate between interlayer and intralayer edges. So we proposed a set of centrality measures that offer this kind of local versus global effect of nodes. So moving away from the biology side, so basically you can think of the multi-layer network as being represented by this kind of a block matrix, where the block diagonal elements are the intra-layer edges, and the off-block diagonals are, off-block elements are off-diagonal elements are the interlayer edges. Okay, and so the overall adjacency matrix is denoted by A tilde, and if you're doing page rank, it's defined with respect to A tilde. But we actually look at both the intra-layer connections separately and the interlayer connections. And then we define multiple notions, right? So first one that we introduced was something called the local centrality, which is essentially a version of page rank. This is the equation for page rank, where P is the rank of the node, and X is the ID for the node, and this is basically the steady state of this equation gives you the page rank. And likewise, you have the local centrality, which is essentially like the page rank, but defined only on the intra-layer nodes. But then we also have something called the global centrality. And the key to this decomposition was that we wanted to make sure that the overall centrality is some kind of a combination of the local and global centrality. And we achieved that up to a scaling. So we show that. But apart from this, we introduced multiple other notions of centrality, where we looked at a global centrality, but targeted to a specific layer, right? Or a global centrality targeted to a specific layer and a specific set. So why did we have to do these things? Because the question that we were interested in addressing was, can we actually identify, something like, say, the insulin producing genes, which are important for tissue-tissue communication, right? So then I'm interested only in targeting those genes in a layer which respond to insulin, and with respect to those genes which are the more central genes in the whole body, right? So that's essentially what we wanted to look at. So we had these different fine-grained notions of centrality that we defined, and the way to read this graph is the x-axis is basically the ranking of the genes produced by our target centrality method, right? And then the y-axis is the recall. So how many of the actual insulin producing genes have we already identified? So this essentially means that, okay, in the top 100, how many of you identified? And so when comparing it against YesSeq, which is the best known centrality measure for multi-layer network that is out there, they have proposed it for a completely different motivation, not for what we are doing here, but that's the only algorithm that was out there, and so we compared against that, and you can see that our method does produce much better ranking of the genes. So basically now you can go in there and then figure out which are the insulin producing genes that we should target. So we started with insulin because we have a lot of data on it, but this can be extended to other kinds of hormones, and we do have other results wherein it has actually thrown up some very interesting candidate genes that could potentially be further validated. Right? So this is the first piece of work I want to talk about. Next is on node classification, right? So where the goal here is that I give you a graph, and for each node in the graph, I would like to assign a label, right? So it could be whether it is an essential gene or not essential, right? So that's one, essential protein or non-essential protein. Or it could be, okay, whether this person, is a computer scientist or not. From a classification, from a citation network, I could do that, right? So the idea here, what we looked at was actually doing this classification of nodes with hypergraphs and with multi-layer hypergraphs. So at that point, this is slightly dated work. When we proposed this, ours was the first method that actually looked at this kind of classification on of nodes on multi-layer hypergraph. So we defined basically a random walk operator for hypergraphs, which is kind of the most natural definition that you could think of, which is essentially the normal random walk from a node would say that you uniformly choose among all the outgoing nodes, all the outgoing edges, and pick one and go to that node. So that is basically one step in your random walk. Right? So what we defined here was, hey, first you uniformly choose among all the hyper edges that are incident on the node. And then once you pick an hyper edge, let's say you pick, so you start from this node and then you pick this hyper edge, then you uniformly choose among the other nodes in the hyper edge. Right? not just uniformly randomly choose some node in the hyperedge, which means you could potentially stay in your location itself. Right? So then we define this random walk, and then based on this, we again go ahead and define a whole inference mechanism that allows you to make inferences on the nodes, like the labels of the nodes. And so you can think of it like it's some kind of a diffusion process that is running, right? So when I start off, let us say that I know for a few of these students, right? I know whether they have a high grade or a low grade or what, right? And then now what I do is using this random walk operator, I kind of diffuse those labels throughout the graph, right? And then determine which of the other people in the graph have a high grade or a low grade, right? So you can think of this as like point knowledge that you have, right? Only for a few points in the system you have the knowledge, and you try to diffuse it and make an inference for all the nodes in the graph. Right? In fact, we have explored using heat diffusion models also for doing this kind of inference. But for this particular hypergraph setting, it turns out that using a very simple slightly modified random walk operator for hypergraph worked really good. And you can see this, just going to give you a sample result. So we ran this on a dataset gathered from Twitter, right? So here the nodes are various entities. In fact, they were the people like sports reporters, sports persons, officials that are involved with Olympics and so on and so forth, and the kind of tweets that they exchanged, right? And so you could look at various relations, whether one is a follower, tweet, and so on and so forth. There's a whole bunch of relations that you can, it's there on the slide, right? That they can define between two people on the Twitter network. And now the goal is, so we have to figure out whether the person was athlete or not. Right? So that is basically, we know a few people who are athletes to begin with, and then we have to basically figure out what the role is, right? Just a toy example that we had. And so the collective ensemble is essentially a way of doing, individually using each one of these relationships, making a prediction and somehow combining their predictions. And that was the best prior result. Right? And so our method, you can see that very comprehensively beats that, regardless of how much information we have about the graph. Even when we have very little information about the graph, and more than 70% of the graph is unknown, we still do much better than the collective ensemble approach. Right? And so this was joint work with two of my master students. So it's Chitosan and Harini. Right? And then the next paper that we worked on, which was very recent work on the same problem, right? How to assign labels to nodes. But here, this is not a hypergraph. This is only a multi-layer graph, right? And we used this following approach. Remember, I had this on the previous slide. I didn't talk about it. So this is an idea called node embedding. So what we try to do here is that we take the graph. The graph has a very large dimensional representation in terms of the adjacency matrix, and then I try to project this to a much smaller vector space, right? So you can imagine, so this representation for this graph in the adjacency matrix space would be a 12-dimensional representation. Now we're projecting this into a 2-dimensional representation, such that some properties of the nodes are preserved in this low-dimensional representation. So this is like an embedding of the nodes into a low dimension. And so what we did here was use some small amounts of label information that was available earlier, right? And did a very nuanced projection using a deep neural network. I mean, I can talk more about it if people are interested offline. So the idea here is that we first start off with, just to show you that it was a complicated neural network, and that's a 5-stage process that we do before we come to the final embedding, right? And then using that embedding, we basically do like a logistic regression on top of it, right? To get the labels. And so what we run it on a variety of different graphs, right? And then and also on compared against a lot of different methods that are out there. In fact, some of these are very close to what we do, except when very minor differences in the algorithm. It just turns out that minor differences were needed to improve the performance further. And again, you can see that we ran on graphs ranging in from very dense graphs, small number of nodes and a lot of edges, to very sparse graphs. And so we showed that we work well across all these settings. And here you can see the results again. And SSDCM is what we proposed. And you can see that in terms of classification, we comfortably beat. I mean, so all the bold numbers are statistically significant results over all the other methods across all the graphs. And then we also tried something interesting. So we looked at what is called node clustering. So if you remember, we are projecting these nodes onto a two-dimensional space, right? So we wanted to see how close in this two-dimensional space these nodes fall with respect to the labels that we are predicting for them. It turns out that the clustering is also very tight, right? And we get much better cluster quality than any of the existing methods, except for one exception. Right, and we can see that when we do the visualization in a 2D space, and each color here corresponds to a different label in the problem, right? And you can see that some of the earlier methods actually are not that great in separating, even though this Han is actually not that bad. But SSDCM actually very neatly separates all the clusters. This is just showing it for one data set. Across multiple data sets, we see this kind of a nice separation of the entities when we do the prediction. Right? Even though they are not, this kind of a separation is not fed to it. This kind of emerges from just trying to fit the label information. So the next thing, it's like I said, there's going to be like a barrage of things. So some work that we did on hypergraph clustering. Remember, the previous clustering work actually used the label information. So that's not truly clustering. I'm just showing you the clustering effect. But here we are looking at the problem of clustering. So what is the problem of clustering? So given a complex graph, can I kind of separate out the nodes into closely connected groups with somewhat weak connections across them? You can think of some kind of modularity maximization and things like that. In this setting, right? So we basically looked at doing hypergraph clustering by trying to match it to this classical modularity maximization framework that you have in graphs for discovering modules in the graph, right? But doing this for the hypergraph. So the normal modularity maximization works like this. So Aij is the adjacency matrix, and Ki is the degree of the node I, right? So the expression for modularity goes as, so you take Aij, which is the ijth entry in the adjacency matrix, and you subtract the product of the degrees normalized by the number of edges, right? And then you use that as the as a score, right? And use this. So you try to partition your graph into different modules such that this modularity score is maximized. Right? So this expression here, right? So where I talk about Ki Kj by 2m is the probability that if I randomly distribute these m edges, that an edge will lie between node I and j, and you will preserve the degree of node I and j from the original graph. That's why the Ki, Kj appear here. Right? So you preserve the degree of the node I and j in the original graph, but then you're otherwise distributing the nodes randomly between the distributing the edges randomly between the nodes. Okay, so we wanted to do the same thing for hypergraphs. So the null model for hypergraphs is going to look something like this. I'm going to look at the null model is di dj, right, which is the degree of node I, degree of node j, which is the hypergraph degree of node I and node j. That's fine, but divided by the sum of the degrees of all the hypergraphs, right? All the nodes, right? Degrees of all the nodes instead of m, 2m, which is easy enough to do in the regular case. Here we actually have to do some kind of a normalization depending on the hypergraph degree of the various nodes. So to get this hypergraph degree, right? So what we did was we looked at a kind of a reduction of a hypergraph to a regular graph. Right? What I mean by regular graph, a graph where each edge connects only two nodes. Right? And the way we derive this adjacency matrix is by doing this kind of a transformation, which we call as a node degree preserving reduction. So it's essentially, it works out this way, right? So for every hyperedge, let's say there's a hyperedge that connects three nodes, then I construct in the graph, right? In the reduced graph, I'm going to have a triangle that connects these three nodes, but the weight of the edges on the triangle is going to be decided in such a way that when I add up the weights of all the incident edges in the node, it will add up to the hyperedge degree of the node in the original hypergraph.
You have consumed more than 21 minutes. Okay.
Yeah, sure. Yeah, I'm also having a timer set off. Thanks for the warning. Yeah. So so this then what you do once I get this, I can use any of the modularity maximization algorithms to maximize the modularity with this adjacency matrix, and that is essentially what we do. And we show that this method performs well, right? I'm actually going to skip the iterative algorithm that we had because that's going to take some time to explain. Again, this is joint work with Tarun, my PhD student, Shankaran, who was an intern with me, and Harini, who was a master student. We already spoke better, and Srini Parthasarathy is a collaborator from Ohio State, right? And again, you can see that. So the iterative method that we proposed does really well. And if you take out the iterative method from the picture, the method that I described, right, the Lumen with the hypergraph reduction that we talked about is does fantastically well, right? And then we looked at this problem. So there's always an issue whenever you reduce a hypergraph to a graph. Right? So you can look at this, right? So this is a one hypergraph with a single hyper edge. And this is another hypergraph with 3 hyper edges. But when I try to do this kind of a standard reduction, right, I basically get this graph with it's a fully connected graph on these 4 vertices, right? So now I've lost some real information, right? Here there were 4 different, there's like 3 different interactions here, but here are the single interactions. I've lost some important information here, right? So we wanted to see if we can come up with a mechanism where we can keep the hypergraph representation as some kind of a tensor, and then work with that without having to do the reduction, right? And then so we came up with this notion of a hyperedge score, right? So in fact, the hyperedge score is essentially if you take the Fiedler vector, which is the second eigenvector of the Laplacian of the graph, right? And then you take the sum of the kth power. If you're looking at a k regular graph, k uniform graph, the sum of the kth power of the components of the eigenvector for that lie on that edge, and the product of those, right? So this is essentially something like the arithmetic mean of the kth power and the geometric mean of the kth power. And the larger this value is, right, then it is an edge that needs to be cut, because there is something very disparate about it. If the geometric and the arithmetic mean are the same, this weight is going to be 0, score is going to be 0, and therefore you can retain it. So what was really interesting for us is that this single Fiedler vector based partitioning, right, seems to capture a lot of information about the spectra of the graph, and we have now been exploring multiple ways in which we can employ this edge score, right, for other things, right, not just for hypergraph partitioning, but also for hyperedge prediction, and also for building a graph generation model, and so on and so forth. And so this one slide. Managed to go next slide. Yeah, there you go. So what we did was we compared it against a standard way of doing graph partitioning, right, by doing a sign-based Fiedler vector partitioning. So that is the RF, and RP is the ratio cut. The cut proposed by our method. Right? So now you can see that in almost all the cases we get nearly 95% improvement over the standard way of doing this using the Fiedler value. Right? And so what I'm going to do in the interest of having some more discussions, I'll skip all the way to my conclusion start. Is it fine, Professor Pradeep?
Yeah, it's fine. It's fine. Yeah.
I actually skipped like 6-7 slides in between so that we can keep the time. So basically, our work has been looking at extending graph-based algorithms to complex network structure, and we're finding that it is not always trivial. It's not the case you take a hypergraph and make it a simple graph and then run your algorithms, right? So we looked at hypergraphs and multilayer networks, and we looked at a variety of approaches. There are a few more which we didn't talk about, in all of these, right, we found that preserving this kind of a higher order information about the interaction is very important. And so we show that we always beat methods that don't preserve this higher order interaction. And so we are extending this to other graph-based algorithms, not just these centrality, clustering, classification, and so on and so forth. And we're also looking at graphs that have more complex structures, such as something called knowledge graphs that's very widely used in computer science, where not just the nodes, but the edges also have types and features associated with them. So this thing can become arbitrarily complex very soon. So these are the various papers I spoke about today. And of course, like I said, I'm happy to talk more offline. Very good questions. And that's a link to all the network graphs related papers that my group has.
Well, very nice. I am sort of somewhat away from this, but let me take a, ask a question, which connects to graphs in general. Chemistry, of course, is full of graphs. Chemical structure is a graph. And you talked about DNAs and things like that. So as the central dogma, central problem in chemistry, material science is structure. And you get structure in bits and pieces and you need to connect them. And one way to get bits and pieces is spectroscopy. And mass spectrometry, one is a very common technique by which you get fragments of a molecule, pieces. Pieces you keep having pieces, but you don't have a structure, because getting a structure means getting a crystal. Getting a crystal is art, and sometimes it takes ages to get a crystal. So you get pieces always, and you would like to go with structure. And this is where graph theory is coming in. But then when you start connecting this and then building structures, it so happens that interactions are very many different types of interactions. Chemists say there is a covalent interaction. There is an ionic interaction. There are, in proteins, there are primary structure, secondary structure, tertiary structure, all kinds of interactions. If you have methods from pieces to construct chemical or material science structures, that would be a theme that would help a lot of people, and which your center can really contribute to.
Sure. So, I mean, this is something which we haven't directly worked on, but we have looked at something adjacent. Okay. So we have looked at generating, drug molecule structures, right? So by using, reinforcement learning and graph representations, and by, starting to do kind of local perturbations, starting with the known molecular structure, start doing local perturbations, and then keep testing it for, validity and properties that you want, and so on and so forth. And then you start producing, drug candidates, like molecule candidates, which can potentially then be, tested out in wet lab experiments to figure out whether those satisfy those properties. So we didn't go all the way to the wet lab part, but we have taken, existing data sets of molecular structures, and then we have tried to, search through those and try to come up with, start with one molecule and move to another molecular structure. This doesn't quite work with pieces of molecule structure, but I'm pretty sure we can do that, because we have done that in a kind of a knowledge graph setting I was telling you about, right? We take pieces of knowledge that we know, and then based on, larger domain that we have seen earlier, right? Maybe other crystalline structures that we have seen earlier, you can start hypothesizing edges that you would want to explore further. I mean, this is, of course, for each one of these problems, you have to work on creating the data set and the training regimen for it, for the machine learning algorithm to work. But certainly, I mean, we can try. I'm not promising anything, but this is a feasible solution path. I can try. I'm really happy to talk about it.
Yeah, of course. I mean, there are plenty of examples like metal organic frameworks, covalent organic frameworks, zeolites, metal clusters. All of them give you a lot of pieces of information. For want of time, I will take this, in case others want to ask questions.
No further questions.
Okay, colleagues, you have something, Rajneesh? Hemachandra, I thought these kinds of things are very interesting for your kind of single molecule structures.
Yeah, no, I don't have any question. Thank you.
We have to tease these computer science fellows.
Yeah, computer scientists are everywhere, but thinking of something very relevant to what we do
They think that anything complex they can solve.
So no, I don't. I'm not saying that, Professor Pradeep. I'm saying the thing to Hemchandra is I can't think of something relevant to you because I don't know what you're doing. Right? The way to do this is have a conversation. Way to do this is have a long conversation. In fact, I started doing some work with Sunny on some material questions. Right? And I'm now doing some. Of course, I do a lot of work with Karthik, and I don't understand genetics. Right? I don't understand much of these, but still we are able to do that because I bring the algorithms to the table. He brings the biology knowledge to the table. So the way to do this is through conversations. I'm not an expert. Certainly not in any of these domains that I work on, right? And so I think this forum is the right place to start these conversations, and we should continue. We just not stop here. I mean, if Hemchandra, if you want to have a conversation offline, I'm happy to do that.
Yeah, sure. Yeah, definitely. Yeah, thank you. Thanks for that. Yeah, I'll go through your things because right now I can't think of something. I know Professor Pradeep just prompted it, but yeah, right now I can't think of something. Yeah.
Ravi, Sanjeev here.
Hi, Sanjeev.
I think I need to talk to you offline, where I guess your graph theory would be quite applicable to find out the side effects of some of the drugs. Can't you hear me?
No, there's some kind of an echo. I'm not able to make out what you're saying clearly.
Is it better? Is it better now?
Yeah, much better. Yeah.
Okay. So what I was thinking, perhaps your graph theory would be quite useful to find out side effects of some of the drugs. So a particular drug which is supposed to interact with a particular receptor,
yes,
many times it interacts with something else.
Yeah. So Karthik and I have explored that a little bit with one of our students. There was a dream challenge some time back, so we actually did some work on that. But we didn't look at the molecular structure itself. Right? It's looking at other kinds of property. Yeah, sure. It's been done. There are few data sets there for drug interactions and drug side effect predictions, and I mean there is some literature out there, and we have also done a little bit of exploration quite some time back. I think the dream challenge itself is about 5 years old now, so we tried a few things back then. Yeah.
Very nice. So we'll move to the next talk.
Thank you.
Yeah, thank you. Very nice and very interesting possibilities. And the next lecture is by Adi Nagarnathan. He is going to share his screen now. Go ahead. As usual, 25 minutes. 20 minutes, I'll give you one.
Yeah. Are you able to see my screen?
Yeah, very nice, and we can hear you.
All right, good. So thank you, Pradeep, for inviting me here and to share my thoughts on this. I'll be talking about protein assembly and function. I'll just give a brief overview of what I have been doing over the last 10 years at IIT Madras. And of course, I do say that working at the interface of physics, chemistry, and biology, I do more of physics and chemistry in a certain sense and less of biology here. And that's because of this particular statement that I'm going to show up here, which some of you might take offense, particularly the biologists. Sorry. Yeah, so I believe at least that biology is chemistry in disguise and it's governed by laws and principles, but with a bit more of right now algorithms and stuff that are trying to uncover these physical laws and principles. A bit discomforting to a certain level to the biologist, but I think it's a great time to be a biologist, as Ravindran would probably agree, because there are so many unsolved problems here, and this is something, some of which I'm trying to address in my group over the last few years. To start off, I'll just show the laser pointer. All right, so I work primarily on proteins, and most of you would know what these are. Proteins are basically the workhorses of the cell, and they play a role in defense. Now all of these antibodies right now with the SARS-CoV-2, they play a role in structure. They contribute to function, communication, storage, like say for example hemoglobin, iron, and so on and so forth, and even for transport, right? So this is something all of you know. But the interesting thing about is this, they're primarily chemical entities that constitute these protein molecules, and these are amino acids. There is a standard structure of an amino acid with an amino group. This is generally positively charged at pH 7. Negatively charged here, this carboxylate group is what you would see here. And the property of this amino acid is determined by this particular side chain, which is shown as R here. And there are 20 different side chains or 20 different amino acids in biology primarily. And they have very different properties. Some of them are non-polar in the sense that they hate water in the conventional sense. And some are polar. They form good hydrogen bonds with water molecules, particularly the termini of these that have either hydroxyls or SH groups associated with them. And some of them are also positive and negatively charged. Makes it very interesting in that sense. They basically cover all the properties that you would expect in a chemical molecule, right? And the convention is that we label them with specific alphabets, and these are 20 in number. And interesting thing here is that you can, just like yes, you make words by bringing together these alphabets. Biology does the same, really, right? In the sense that, or with the help of chemistry, in the sense that you have a peptide bond that is formed when you bring two of these amino acids together by releasing a water molecule. These are carried out by enzymes within the cell. You can also do this in vitro, outside of the cell, using chemical synthesis as well. And once you have these polypeptides, and you eventually start adding one after the another, and you bring together these big polypeptide chains. All right, so this is effectively equal to the sentences and words and paragraphs that you have in a particular book, right? So these words, the letters, they come together, form the words, and the words form sentences, sentences form paragraphs, and books, and so on and so forth. This is exactly a similar organization that happens in proteins. But the unfortunate thing when it comes to this particular field, which is basically protein folding, is that not many that goes into these building blocks coming together, right? Why should G follow F or W follow P, for example? That grammar is not pretty obvious, and makes it quite challenging, and that's what is the protein folding problem. And I'll come back to that in a second. So what is known is that, you already know, as Pradeep also alluded to it, that you have a primary sequence, which is basically these set of letters that are come together forming this polymeric chain. And these constitute some kind of code that directs the protein sequence to fold into a specific structure. And of course, this structure contributes to some function, which some ligand binding and some protein-protein interactions, or protein DNA interactions, for example, right? And these things, interactions, and even the chemical composition vary with time, and this contributes to evolution of the sequence, and hence the structure, hence function. And this goes back and forth, and this is happening as we speak right now. Right, so, and this is what I discussed. But now, over the last few years, last 15 years or so, what has happened is that a lot of work has been done on not just these so-called folded proteins, but people are also interested in something called this disordered polypeptide chains. And it has been noted that almost 30% of the proteins inside a human cell is actually something that does not samples a very ordered structure the way you see here. All right, so majority of them are actually disordered like this. They just fluffy, moving around within the cell, making these random interactions, and maybe probing and figuring out whether to bind to something or not. Right, and this effectively significantly heterogeneous in terms of their conformation, and a lot of functions that are associated with this. So you have this structured protein, you can also have this so-called intrinsically disordered protein, IDP. And even for a well-folded protein, you can have a disordered tail that probably mediates interactions. And these disordered regions can fold either with themselves or in the presence of another binding partner. So there's something called the templated folding, as some call it, right? So this is some aspect that has come up over the last 20 years or so, 15 to 20 years, but there are additional layers to it, which I'll also briefly touch upon over the course of this particular presentation. Right, and so now we know the sequence and we know the structure. So what is making it very difficult to address this particular problem is that if you consider this any polypeptide chain, an unfolded protein can sample an infinite number of conformations, which Leventhal kind of showed in his back of the envelope calculation. And also, for example, the simplest way to think about this is that if assuming that each amino acid can sample 3 conformations, then of course for 100 residue protein, you will have about 3 to the power 100 conformations. And even if the polypeptide chain is able to sample 10 to the power 12 conformations per second, it will take more than 2 to the power 20, 2 into 10 to the power 28 years to actually reach that particular folded state, or identify that particular state that corresponds to this beautiful structure that I showed you before, right? So, and this is not the case. So, which means that if you think about it, you can imagine a hypothetical landscape here, a conformational landscape, as we call it, or an energy landscape. So, and you have this ball that is randomly moving moving around on this particular state space, right? And it's trying to identify this folded state, which will never happen if there is no real driving force towards the folded state, right? And what people have done by linking together physics of polymer systems and the condensed phase matter theory is that they have come up with this so-called energy landscape theory that suggests that a protein need not sample all the conformations, and it actually kind of flows hypothetical funnel include landscape. So all the molecule has to do once are you able to hear me? Sorry,
Maybe you can repeat the last sentence.
Yeah, so I got a notice here saying that my network is unstable. So effectively what happens is that this landscape, hypothetical landscape, the way we want to see this, seems to direct the protein molecule which is unfolded, that can be, let's say, at the edge of this particular funnel, to actually fold to the native state. Here it seems to fall out, but imagine it's going here, and that's what this is. So these are effectively landscapes that one can derive by actually sampling these conformational states. So how does one do that? And what is the problem, right? So particularly when it comes to protein folding, there are 3 different questions that one could ask. And these are just very broad questions, and I will not go into the details here for some of them. So the one is that, can we predict the protein structures given this one-dimensional string of amino acids? So you have this genome sequencing method, so you can actually read all the bases that are present in your particular gene, and from this gene you can actually translate it into a amino acid code. But there are no real methods that are currently available that can consistently predict as to what will be the final structure of this particular sequence, right? But as you would have noticed over the last couple of days, there is this two of these papers that are published. One by the David Baker's group in Science, which is Rosetta Fold, and this is the Google group that publishes this Alpha Fold, one in Nature and one in Science, where they have come up with machine learning methods to actually solve this problem for sequences whose structure are not really known. So they've been able to do it consistently, purely without understanding the physics or the chemistry, but purely by learning from the known connection between sequence and structure. All right, so I'm not going to touch upon that. Maybe we can discuss that later. The next question is, what are the mechanisms of protein folding? Now you have this polymeric chain. So what are the different routes does this polymeric chain takes to fold into the native structure? For example, you can imagine that you're starting off from the IIT main gate and you want to reach the Besant Nagar beach. How many different routes are there for you to reach the Besant Nagar beach? Of course, you can say that there are probably infinite number of routes that you could take, depending upon the number of different steps you take, which direction you go, how much you backtrack, what are the signals that you have, which are effectively free energy barriers, for example, right? All these are the interesting questions at a fundamental level that one would be interested in. And this is what I am interested in when it comes to mechanisms of folding. And of course, there is this related question of how and why do proteins fold so fast? Simply because they do so in the matter of few seconds to sometimes even in the matter of microseconds and milliseconds, while technically if these polymeric chains are randomly sampling conformations, they will take infinite amount of time. So trying to connect the polymeric property of this chain to this chemical nature of these amino acids to come up with generic rules that says, okay, if you have this sequence, if you have this particular chemistry that is associated with it, what would be the final structure? What will be the mechanism? How many different routes are possible? And these are the questions that we are primarily interested in our lab. All right, so and just kind of summarizing here, we do a lot of experiments, primarily spectroscopy and chemistry, spectroscopy and thermodynamic measurements and calorimetric measurements, and we do also.
Again, we lost you, I think.
Theoretical modeling and we kind of come up with self-consistent measures of folding conformational landscapes. And with this in hand, we ask some fundamental questions on barriers, coordinates, excited states, and the constraints that a polymeric chain has to face while it's actually folding, and the role of conformational entropy in energetic degeneracy. And of course, something which we are addressing for the last few years is on heterogeneity in folding, binding, and function. I'll briefly touch upon some of these, and maybe we can discuss later and maybe in question session as well. So the questions are, can we come up with some unified model of protein folding mechanisms? And I did tell you that there are certain proteins that do not have any specific structure. They are called the intrinsically disordered proteins, IDPs. And what are the features, what makes them intrinsically disordered, right? And also something we are interested in is how do protein act as sensors of ambient conditions. There are a lot of proteins within the cell that sense changes in calcium concentrations to temperature, to pH, and so on and so forth. Anything that you can imagine, that is a protein that senses it. So how are these sensory features imprinted on the structure of a protein, and hence the conformation or the ensemble of conformations that the protein populates? Right. So this is an interesting question that many of us are working on. And of course, again, these are questions which I don't have an answer, but what are the protein and protein DNA assemblies that control bacterial infection and hence sensitivity to drugs, and this has implications in multi-drug resistance and so on and so forth. And answering this first question, if there is a unified model, can you come up with a unified model? I believe we have some kind of an understanding of a simple and rapid quantitative measure of folding landscapes of proteins. And this is something called the Waco-Saito-Munoz-Eaton model. And Waco-Saito independently developed this independent of Munoz and Eaton. And they developed this in 1978, or Munoz and Eaton developed this model in 1999. And the idea here is that you take every residue or the bead in this polymeric chain that you saw. It's considered to sample only two sets of conformations, one that is 0 and one that is 1. And which means that for a 3 residue peptide, for example, you can have 2 to the power 3, which is about 8 conformations that you see here. So if you know the structure of this, say for an NMR, for example, then you can actually kind of subsume the structural information in terms of basic thermodynamic parameters, and hence you will have the free energy of each of these states. And once you have the free energy, you could calculate the statistical weights or energies if you want. And from the statistical weight, you could calculate the partition function. And if you have the partition function, you would be able to predict majority of the thermodynamic properties that you're interested in, and even kind of sample these state space by doing these random walks that Ravindran was talking about. Here, you will be weighted by this particular Boltzmann's weight, and that will direct you in one way or the other, depending upon whether a particular state is more energetically favorable or less energetically favorable. So this is a very simplistic model that was developed in 1978 and 1999. But what we have done over the last few years here at IIT is that we have kind of taken this model and we have added several different force field terms into it. And that includes van der Waals interactions, electrostatic solvation terms, basically the effect of solvent. You also have some kind of color to the model that comes from the conformational entropy of the main chain and side chain. And you can actually simulate the effect of salt, pH, temperature, and denaturant effects. You can also simulate the effect of single point mutations. The correlation is not great, but this is something still pretty good compared to what any other server or a model can do. These are predictions coming from those residues that are buried within the protein chain, the change in stability of these particular residues. You can also have approximations of this model, and you can derive folding landscapes similar to the one where I showed you this ball that is rolling on the surface. And you can also use this to study protein and protein DNA binding as well. Again, I'll touch upon this briefly. Some applications are, for example, this is I-kappa B alpha. This is something called the repeat protein, and you can see this particular unit that is shown in blue is kind of repeated about 7 times out here. And this has a lot of role in cellular conditions, and is supposed to be highly promiscuous, meaning it binds to several different partners and regulates function. So one of the questions was, how does this regulate so many different functions? And what we did was we characterized this particular protein with this WSME model, which is the Waco-Saito-Munoz-Eaton model. And what you can see here is that we can show that as a function of temperature, the protein is increasingly sampling several different states. And this is similar to what Hemachandra would get from his single molecule measurements, for example. And once you have this, you can actually project these conformations onto a 2-dimensional landscape with 2 different reaction coordinates, let's say, and the y-axis being free energy in kilojoules per mole. And what you can see is that, and the protein does not sample one specific state, but it seems to kind of really move around, sampling many different states driven by thermal energy and thermal motions. And this is something we termed as the domino-like destabilization mechanism. Simply coming from the fact that if one repeat is destabilized, what happens is that this interface is lost, and hence this repeat is destabilized, And because this repeat is destabilized, this will also be destabilized, and so on and so forth. This is very similar to a domino, and we call this a domino-like destabilization mechanism. We have used this model for about 40 different proteins, and this is just one observation that I'm showing you. And linking what Ravindran was talking about in terms of this graph theory, just as you would imagine, a protein is not one specific conformation. Whatever you see in your chemistry textbooks or biochemistry textbooks or in these talks, even the ones I showed you, are effectively averages. In reality, what the protein does is it samples so many different conformations, millions and billions of them, and which means that just the question I asked you was how many different routes are there for a protein molecule for you to go from the Adair gate to the Besant beach. Similarly, a protein molecule also faces this conundrum as to how many different routes are there, what route do I take depending upon what condition it is, right? So, and it's not obvious. So what we did was we were able to map out this conformational states by using this explicit energetic and entropy descriptions that we have modeled. And we were able to connect similar networks. This was done with the help of Sayan Rano, who was at the computer science department, but right now he's at IIT Delhi. And you can see here.
Can you please switch off your video?
This is effectively a node that corresponds to one specific structure of a protein.
The connection is very weak.
Sure. So the internet seems unstable. Yeah, 1 minute. So, are you able to see my slide?
What is computer science doing about network connection?
We are not able to see your slide, Asti.
Now?
Yeah, not yet.
Okay, not yet, is it? Okay, give me a second. Now?
Yeah, you are good. Yeah.
Good. All right. So similar to what Ravindran was saying, so here we have constructed a graph theoretic network. This is just about 1,000 nodes. You can imagine you can even construct this as millions and billions of nodes. But of course, we don't have the computational power. Rather, you'd also see a significant degree of degeneracy associated with these parts. And so what you can see here is a polypeptide chain that starts from here. It could either take this route to the folded state, or it could take this route to the folded state, or it could take this route, and so on and so forth. And we just did this not for one protein, but we also did for several different proteins. And really, you see this beautiful complexity that emerges in this particular system simply coming from the fact that we married concepts from chemistry and physics to construct this statistical mechanical model and combine this with graph theoretic approaches that basically is a computer science based approach, right? So to come up with these nice pictures that really depicts, if it's some kind of take home message that you can take is that a protein does not sample, or rather take one route to the folded state, as you would see in a chemical reaction where a specific bond is formed and another is broken, for example. But in this case, there are so many different ways that the molecule can take to reach the folded state. Right, we also quantified the numbers. I wouldn't want to go into this, and effectively the numbers can vary depending upon what temperature is this, what stability conditions it is, what kind of clustering you do, whether you do a Markov clustering or secondary structure based clustering, and so on and so forth. Right, so and this one particular computational approach that we take, and from the experimental side of things, what is known is that this is this small protein that we have worked on in the lab over the last
consume 20 minutes
already. All right. Yeah. So, but I'll anyway quickly go through this. So what we have shown here is that this particular protein samples so many different states. And one of the predictions coming from the model was that the protein also samples the fully folded state. And we were actually able to predict this, or rather validate this particular prediction via experiments, simple thermodynamic measurements and kinetic measurements. We were able to do these particular predictions and still validate them. And we also had interesting observations where the protein, since it's a DNA binding domain, it needs to have these lots of positive charges right next to each other in the close to space so that it can bind the negatively charged DNA, and which means it's a significant frustration in this particular system. And we were able to actually tune this frustration by adding salt, and you can see that the disordered protein can be made to go to completely fold by adding salt to the system. And this resulted in an interesting mechanism that we proposed, which is called the continuous conformational selection mechanism. Is that fact that when you have a DNA molecule and the protein is not next to the DNA, it's completely unfolded, but when you move the protein molecule, when the protein is randomly sampling states and is attracted towards the DNA, for example, at one specific point, what you will see is that the protein actually increasingly samples folded like conformations in this particular landscape. So this is something which we propose is a novel model, which is something called the continuous conformational selection mechanism of protein DNA binding. And another aspect that we are working on is on mutations in proteins. Again, I'll briefly go through this. The observations that people generally make is that when you have a specific residue and you mutate it, it will only be the first shell of residues that are affected when you make this mutation. But it's obvious that when you affect these particular interactions between this residue and this particular residue, you can also imagine that the interaction between this residue and these residues will also be affected. So imagine dropping a pebble into a still water and you will see this ripple. Similarly, what we think is happening when you make mutations in proteins is that the effect is not just localized, but kind of spreads out and into the structure. And we validated this again via molecular dynamic simulations and network analysis of protein structure and reanalysis of experimental data coming from NMR chemical shift perturbations. And I skip this for the interest of time. And how do proteins act as sensors of ambient conditions? This is a two-component system that we have in E. coli, where these two proteins actually interact at specific conditions and regulate the expression of genes, particularly virulence genes in enterobacteria that infect human gut. But higher temperatures and higher ionic strength, which effectively corresponds to the mammalian conditions, what happens is that these two proteins are not able to interact within the E. coli, and this results in the expression of virulence factors in the genes. So how does this happen? How does this particular protein CNU sense that the bacterial host bacteria has infected a host and it's able to change its conformation? And this is something we were able to prove by a combination of spectroscopic measurements and simulations and perturbations to show that this particular helix that is located in this protein samples very low phase space at low temperatures. But as the temperature is increased, it samples increasingly disordered conformations. And interestingly, this is this particular region that binds this partner HNS. So effectively at higher temperature, this interface is not well defined. So this complex is unable to form, and hence it's unable to bind to DNA and regulate expression. So this is just an example. And we also use this as a sensory protein for ionic strength and pH and so on and so forth. And I'll skip this part. And the future plans in my group, if anyone is interested in collaborating or contributing to it, is to study protein-protein and protein DNA complexes from the perspective of the model, to understand concepts related to allostery and particularly to liquid-liquid phase separation that has completely taken over biology over the last 5 years. So effectively, you see droplets of proteins and protein RNA that you see within the cell. Not just freely diffusing proteins, but droplets you see within the cell, and which means that you need concepts from physical chemistry and imaging to address these liquid-liquid phase separation. We are also interested in engineering enzymes, something we are currently doing, and to also understand the multi-drug resistance at a very basic level. And I'll stop my presentation here, and I thank all my students and collaborators and the funding agencies. Thank you.
Very nice. I would ask like others to ask questions.
Adi, they are here.
Yeah.
Can you hear me?
Yes, yes.
Yeah. Okay, yeah. So very nice talk. So the question I have is, you talked about this various folding pathways for the protein.
Yes.
Are you also thinking about calculating rates of folding in these pathways? Is that in your mind in the future?
Yeah, the rates, the problem, there is no real connection. I can say the way we did this folding pathway calculation was that you have the state space, and we randomly sample using a simple Metropolis criterion and Monte Carlo simulations, these different states, right? And all we have is this MC steps here. And we do not have a direct connection between this to molecular rate theory unless we do some kind of stochastic simulation here. But something we haven't done or explored here.
Okay, so because there are a bunch of techniques available, which I have come to know when I was a postdoc in Amsterdam. So there are transition path sampling theories. There are a bunch of them.
Yes.
Once you know the order parameter through which this protein is folding, you could actually devise a method to calculate the rates That has kind of evolved quite a bit in the recent times. So I was wondering if you could.
Okay, great. So, but would you be able to calculate the rates for each of these routes?
Yeah. Since you have already mapped out the order parameter along each path, I think that would be quite possible, because there one of the challenge there was to know what is the order parameter through which your protein folds. So now you have figured that out, right? You already know that. So now having all these known pathways, then there are very cool techniques out there, which this David Chandler and Peter Bolhois, these guys actually worked on his transition rate theories. Maybe I'll make an appointment with you and
Definitely. Yeah. Please drop in an email and I'll be happy to talk to you.
Thanks, Adi. Yeah.
Thank you.
All right. Are there other questions?
Okay, can I go, Professor Pradeep? This is Chandra.
We have time. We have 3 minutes left.
Sure.
Okay. Hi, Adi. Very nice talk.
Thank you.
So this is regarding your DNA CITAR, the model that you have proposed.
Yeah.
So I'm just wondering if you see any sequence-specific information for, you propose that as the protein approaches the DNA, it starts folding. So there should be some kind of hydrophobic collapse or something that probably needs to happen for the protein to fold, because it's approaching the charged residues, right, or the DNA basically.
Yeah.
So do you think there is any sequence-specific information that can be obtained in the sense, like just to identify what kind of mechanism is it? Really the hydrophobic collapse that is driving the protein to fold as it is the disordered region to fold as it is approaching DNA, or you think any kind of repulsion activity, because you have a negatively charged DNA there and some kind of repulsion is driving the disordered region to fold? And any insights on that?
Yeah. So effectively, what you would see here is that what we are saying is that, this was our original proposal, that the protein is disordered in solution. But when it approaches DNA, and we propose that the protein is disordered because it has all these positive charges that are close together in space, and the protein kind of struggles to bring this together because of the significant repulsion. Right. So, but the moment it's within this electrostatic field of this particular DNA molecule, the field actually shields the positive charges from seeing each other.
Okay.
So, and our initial proposal, I'll just briefly tell you what happened really. So, our initial proposal was that it's primarily driven by electrostatics.
Okay.
So, but when we actually looked into the sequence, I'll just go here. So when we actually looked into the sequence, and what we found was that the protein can be made to fold. This is the same protein, and what I'm showing you here is temperature and a measure of secondary structure along the y-axis here. And what you can see here is that the wild type of this particular protein is disordered, meaning it has less negative signal here.
Right.
But when you introduce these two individual hydrophobic substitutions, alanine to valine, which is a small hydrophobic residue to a larger hydrophobic residue,
right.
or this to this, the protein still remains disordered.
Oh, okay.
But the moment you introduce this mutation, A29V and A48M together, you see some kind of emergent behavior where the protein molecule collapses and folds, even at the same conditions when there is no DNA.
Okay.
So basically there is some kind of selection both at the level of hydrophobicity and also at the level of electrostatics for this particular protein molecule to be disordered in solution. We also had NMR experiments that we have currently done that shows that the protein is actually even disordered within the cell and in the presence of the entire 300 residue multi-subunit domain.
Okay.
And so effectively this is a very convincing display is what I would say. Yeah.
Okay, thanks. Thank you.
Thank you.
Professor Pradeep, is there any chance for?
Yeah, you have a minute or so, maybe.
Sure. Professor Adi, this is just not like a question, but like just like a thought. I just would like to ask you. So, when we are considering these protein conformations, so can we consider them as like similar to the quantum mechanics where you have this basis states in order to expand the entire wave function? So do you think that we can use these conformations as the basis in order to represent the true nature of the protein so that like you are optimizing the coefficients when you are expanding the wave function? Do you see any?
Yes, I mean, you can imagine them. You can use the same discrete state representations. Of course, you cannot, you would have to imagine if you use a continuous variable, it really becomes difficult. Maybe Ithiraja can probably answer that particular question better. But here, So sorry, I'll just briefly go through this. Yeah. So assumption I make here, for example, in this case, is I'm still assuming that there are only these discrete states that are possible. And this, for example, this will be a fully folded chain, and this will be a fully unfolded chain, and you can have combinations of ones and zeros here. And these will be the different energy levels. This will be the lowest energy level. This will be the highest energy level, and the population across these energy levels, when you change temperature or pH, the population across these energy levels is what is changing. So we effectively have a similar quantized description when you have this statistical mechanical treatment. But unfortunately, it has its own limitations, obviously. But molecular simulations, like what Hydrogel will talk, is probably the way things need to be addressed, where you have a continuous description and you monitor things as a function of time.
Okay, we will take this offline then subsequently.
Yes.
We'll go to the last speaker. Thank you very much, Adi. Hidayah Raja. And as always, I will give you a warning after 20 minutes. Good, not full screen yet, but
yeah. Can you see my screen now?
Yeah, everything is okay. Right.
And am I audible?
Yeah.
Okay, good. Okay. Okay. So first of all, thanks, Professor Pradeep, for organizing this nice initiative. And so, although so I'm the last speaker, and the time is very limiting, and we are just I'm in between you and the dinner. So I will keep it very brief. And my purpose of this talk is to just give an overview of some of the problems that we are doing in our labs. And you will see that the subject is quite interdisciplinary, and I will show some examples where we study systems from chemistry, proteins, from bio, and colloids, and etc. So I hope that would be interesting to some of you. So, so this topic is looking at colloids, and colloids are loosely defined as objects which are in the size range of 1 to, let me, okay. Yeah, so basically objects which are between 1 nanometer and 1 micrometer, that's loosely defined. But if you look at the objects that we work with, there are very many systems that falls in this size ranges. For example, surfactant micelles, unilamellar vesicles, mini emulsions, nanoparticles, various macroions, even globular and structured proteins. And why these are interesting? Because you could actually describe the dynamics of the systems using classical mechanics. So you don't really need quantum mechanical description. So you could solve Newton's law of motion for these objects. And they are not typically subjected to inertial effects. So they are mostly Brownian. They are thermally actuated. Thermal energies are important. And the way they assemble, aggregate together has to do with the way they interact with each other via physical interactions without any chemical changes. It also allows us to tune these interactions via changing external conditions like pH, temperature, salt, ionic strength, and so on. So that's what makes this this body of objects as colloids as an interesting system to study. They are big enough that they are you can actually see them in real space microscopy, confocal microscopy, or optical microscopy. At the same time, they are small enough that they are subjected to Brownian motion, and you could also tune the interactions. So because of these features, people actually associate colloids as big atoms, because you could make them assemble, crystallize. You can make gels out of it. You could, whatever you do with atomic system or molecular system, you could very well do the same thing with colloids as well. So now, if you look at traditionally what people study in terms of colloidal interactions, so one of the basic thing is having a hard sphere interactions where two particles can just be apart. Whenever they touch, they basically get repelled because of the excluded volume. So when you have such a very simplistic interactions or you could actually predict that such a system would undergo fluid solid transitions. This was initially predicted from molecular simulations, but later on found by experiments as well. So we can see that if you change the volume fraction of such colloids, you go from fluid to crystal, with an intermediate transition between fluid and crystal. But then in real system, we see that there is also liquid phase in this suspensions. So in order to explain that, then people talked about various other interactions such as DLVO interactions, where you have a long range electrostatic repulsion and short range van der Waals interactions. The potential goes something like this, that when they are very close to each other, there is an attractive well, and then there is a repulsion because of electrostatics, and there could be secondary minima also. And so later on, people talked about various other type of interactions like depletion interactions, which was originally developed by Asakura Osawa models. And then also we have this dipolar chains particles with polarity. So these are very classical way of looking at colloidal interactions. If you look at any colloidal textbooks, you will see particles with these interactions being studied both in theory, experiments, simulations also. But what has happened in the recent times is that we have actually gone beyond these classical interactions, and we have done a lot of, we in the sense, people in the literature. So they have looked at a lot of fascinating ways in which you can do surface chemistry on these colloids and then bring a lot of new interactions. One example is what is making a lot of big news is this DNA-coated colloids. So you start with a very bare, uninteresting colloids like polystyrene or PMMA colloids, and then you graft on the surface a DNA with the sticky tip at the end. So these are basically single-stranded DNA, and then you could have another particle, same particle, but coated with a complementary DNA. And because of this pairing, DNA pairing, you could actually tune the way that the particle would interact. And the temperature is a very nice control variable where you can actually make them click or not. Okay, so that's very nice. And the people have done a lot of work on this DNA colloids, making fascinating crystals, multi-component systems, and so on. And another idea is looking at this, what's known as this patchy colloids, where you basically surface engineer partly on one side of the particle, on both sides of particle, where the middle part, so for example, you could say that the middle part is charged and the opposing poles are hydrophobic. Okay, so you're basically making a model very similar to a protein, which has this patchy charges and patchy hydrophobic entities. And there are other fancy things called the classical critical Casimir colloids, which are slightly difficult to control because here we often have a binary mixture of liquids. And when you disperse particles in a binary mixture and then you play with your temperature, you could make them attract or repel, which is very tunable. So why I'm saying all this is because it seems that in the recent times, we are able to make a lot of advances in tuning the interparticle interactions, bringing in biology, chemistry, to this colloidal science, thereby create a very wide set of tunable interactions. Thereby, we can create a lot of interesting structures out of these colloids. And the idea here is to make or build new self-assembled structures, macroscopic structures, which could be used as a functional materials of various kind. So with this background, so I would like to basically motivate. Pretty much our research is driven by the following, that we recognize that when you look at molecular assemblies like surfactants and proteins or polyoxide metal assemblies, so these are basically driven by there's a shape anisotropy, there is a chemical dissimilarity, multi-component systems, and the interactions are site specific. And of course, thermodynamics drives these systems from to equilibrium. So when you have these things known, so the question is, can we impart similar anisotropic shape, chemical dissimilarity, and site-specific interactions on a colloidal scale, which is a larger length scale compared to molecules, and then thereby mimic structures of what is known as colloidal molecules, exhibiting a very similar structure like a molecular assemblies. And the reason to do that would be, we could think of having a larger assembly for applications like drug delivery, photonic crystals, or membranes for separation, etc. So that is the motivations. And then if you look at literature, you will see colloids being synthesized in various forms. You got this, spherical particles, and then you can actually make a compartmentalized particles based on polymers. So different color means different types. For example, here you can see hydrophobic, hydrophilic part, which could be a spherical or rod-like particles, or there could be discs, and so on. And or you could make a surface engineering so that you make a part of the particles patchy with a different type of interactions. And there is also anisotropic shapes, which there's a lot of development that have happened in the recent times in making such colloids in the literature. So now, so coming to our research in the last 5-6 years or so, so we have been looking at the various means by which we can create this, this different type of patchy colloids. So here is something I'm just trying to highlight here. So what you see here is a pointy single patch on a substrate particle. So here in the backbone is your silica particle, and this is basically a polymer patch. And the polymer patch can be made hydrophobic. So when you keep these particles in suspension, you can actually specifically arrest the formation. You can specifically induce the formation of dimer, because the patch will attract and stabilize the dimer-like structures. Or you can make multiple such patches on the equatorial plane. In this case, there are some 6 such patches using some of the interfacial techniques that we have developed. Or you could even make a metallic patch, make a gold patch on a polystyrene. And this gold could be used to, again, hydrophobicize or attach an antibody for a sensing application. So here we show that the same gold is basically patterned for a different patch coverage. So here you have made very small patch coverage. Here you can make a much larger patch coverages. We also go beyond spherical particles. We make this kind of idli, what we call idli shaped particles, or this kind of a faceted particle. And these things are very easy to do because you could actually make these things using interfacial engineering. Let me not go to the details of how we make these particles, but I will just highlight some of the work we have done with these particles. So recently, we were actually looking at some making of composite particles by bringing nanoparticles and colloidal particles together. The idea here is that generally, when people work with nanoparticles, we can make nanoparticles, we can study them, but then when you want to make a functional device, or when you want to substrate with this nanoparticle, it becomes very difficult to assemble them uniformly. So one of the way we thought we could solve this problem is to first assemble these nanoparticles on a colloidal particle. Then later on, as a second step, you can actually take this microparticle and then assemble again into a larger substrate, because manipulating colloids are much more easier than manipulating nanoparticle. That's what our experience has been. So in that context, so if you see here, what you see on the background is this is a polystyrene particle, and then we make a ring of gold spherical particles being assembled as a ring here. And we demonstrated that you could do that for other particles. For example, here we assembled hematite ellipsoids, and here we have assembled gold nanorods on the substrate particle. So this we do by simple dip coating, where we first make a monolayer of polystyrene particle spheres here. When I say colloids, I'm talking about micron-sized colloids. And then you have a suspension here, which has gold nanorods. So what you do is, in a controlled way, you basically dip coat this particle in a substrate. And then you basically, when you dip coat, you get this kind of wedged film. And within the film, you got this gold nanorods. And then by controlled drying, evaporation of this liquid, and then you rip off this particle away, then you basically get these nice rings that are shown here. And you can do it for a variety of particles. And we have demonstrated that it's quite robust in in doing this. This is one nice piece of work that we have done recently. And this is another type of particle, which we call the spherical cap particles. So we actually start with a spherical particle and do some surface engineering so that it basically changes to a or a spherical cap-like particles. This is very interesting because there is a lot of changes that happens in the surface chemistry when we change the shape also. And because of that, we could actually assemble them as a linear chains, which we call staggered polymer chain. So as you can see that a dimer, trimer, which very nicely assembles into a linear form. And this is basically a SEM micrograph of a dried sample. And these are basically the in solution, the structure are imaged using optical microscope. And these are not something that happened because of drying, because I can show you here by this video that it is actually, let me just play this video. So you can see this nice Brownian motion of this long chains. You can also see smaller aggregates on the backdrop, which are also in thermal motion. So quite reproducible, and you can get a distribution of lengths. And we have an explanation why this happens, because of the way the interactions are tuned, which is basically electrostatic and van der Waals forces. Because of the shape asymmetry, we can actually play around with this interaction so that they can assemble always in this kind of chain-like fashion.
What is the scale bar here?
So these are about, so these particles are about 2 micron in size. Okay, so this is about 2 microns. Yeah.
Carry on.
Yeah. Okay, so we could actually go a little beyond because there's been a lot of interest in the recent times on active particles, which are basically mimicking the motion of bacterial systems. To do that, what we did was we took the spherical cap particles, and then one half of this on the one hemisphere, one side of the hemisphere, we have a coating of platinum, and then you can actually disperse this. As you can see, this the platinum coating with this contrast here. You can see here also. So this rough patch that you see, sorry, the rough patch that you see here is basically platinum, and that's a bare surface. And the reason why we do platinum is because when you put this particle in a solution of hydrogen peroxide, so basically there is a reaction that decomposes hydrogen peroxide into water and oxygen. And due to this, there is a chemical gradient of chemicals that gives rise to diffusive phoretic mobility, and you can actually make them move. And that's this slide, where you can see that you basically create a variety of motion. Okay, is it moving? Sorry. Yeah, so you can see that you basically, depending upon the amount of hydrogen peroxide or the fuel in the system, and depending upon the orientation of this particle to the substrate, you could either create a kind of a linear motion or a very circulatory motion, quite repeatably. We have done with a lot of different type of particles. It's quite reproducible. This is very nice because it kind of controls. Suppose if you want to attach a drug molecule to this particle, you could either take from one location to another location, or you could create a local circulation because of this. So a lot of nice things one can do with this. Let me move on with this. So again, there is again explanation why this linear motion versus circular motion happen, depending upon the orientation of the particle in the substrate. Maybe in view of time, I will skip that mechanism. I can discuss that maybe during Q&A. This is very nicely, this dynamics could nicely be captured using classical Langevin dynamics. So basically you have a particle which is subject to thermal noise. At the same time, it has a self-propulsion velocity. So you can set up a Langevin equation and analytically solve, and then you will predict that the mean square displacement should be given by this equation. It's linear in time as well as quadratic in time by the two different time scales. And all these data points and the curves are basically fits and experimental data, and it very nicely explains this classical Langevin dynamics problem total. Also, you can also look at the circulatory motion that I was talking about. So this also, again, you can analytically solve this problem using Langevin dynamics. And you see this what we found experimentally on the mean square displacement could very nicely explained by this theoretical model that is out there. All right, so moving on. So I'll just skip this because this is basically reproducing the same thing with a different particle size. Okay, just to give a moment. So basically, when you change the particle size to a smaller scale, the Brownian basically kills this orderly motion. So that's what you see, that when the particle size is about 0.5 micron, then you basically have very chaotic motion, and all the circulatory motion is very local and not very persistent. So again, this is basically very reproducible and was shown for two different particles of different sizes as well. Okay, so moving on, I'm going to a slightly different topic, which is something that I did a very long time ago. Is to do with self-assembly of polyoxometalates. This is basically oxides of molybdenum and iron oxide, which nicely forms 2 nanometer to 3 nanometer sized spherical objects, very highly ionic in nature, soluble in water. And then when you look at this solution of this polyoxometalate, people have found that it forms a nice unilamellar vesicles. So at the time, there's a lot of confusion about why this happens to be, because this seems to be a spherical objects, and stabilizing a hollow shell like this was very a mystery. So we were looking at what is the reason for such unusual self-assembly. So when we looked at the crystal structure of this polyoxometalate, so we found that there are hydrogen bonding centers that comes from this iron oxide centers. So we basically coarse-grained this polyoxometalate chemical molecule into a patchy model, where we basically put attractive patches mimicking the hydrogen bonding wherever the iron oxide centers are positioned. So that basically gives a very regular icosahedral symmetric patchy model. So rather 30 patches. So we do Monte Carlo simulation of that, and then show that these molecules spontaneously form this sheet-like structures. Of course, I mean, at that time, we couldn't do a very large system simulation, so we did a very small scale system. And we showed that a fragment of this could show kind of a planar structure. So if you turn it around, you can see this single sheet-like objects. And so you can basically expect that when you have a very large system, they basically become very large sheets, and then they eventually fold to form this polyoxymetallate vesicles. So again, the reason for that is that so because of the symmetry that is involved, depending upon which way these sheets grow, you have energetic advantage of one pathway over the other. For example, if you look at this perpendicular to the C2 axis symmetry, this center molecule here could actually bind with 6 neighbors, making a hexagonal lattice. Whereas if you look at this perpendicular to the C5 axis, you basically have too many patches. There are about 10 patches here. And because of this excluded volume, you can only make a 5 folded packing on this in this. So because of this energetics difference between the number of neighbors between these two types of orientation, the particle tends to grow in this hexagonal sheets rather than growing in this way. So I will also skip this because it's basically more details to show why this forms 2D sheets. In fact, if you go to slightly larger concentration, you can also demonstrate that not only you get one single sheet, but the single sheet also makes a multi-layer sheets. And this is also experimentally shown that when you age these vesicles to longer time, you also get a multi-lamellar vesicles. That also points, the precursor of that is also shown in experiments. I'll skip this. Yeah. And another project that we worked on recently using this patchy model is looking at gelation of polymers, PAA, polyacrylic acid. And when this polymer is water-soluble polymer, when this polymer is actually mixed with guanidine, and then you change with the pH, you could actually change the structure of this polymer solution from a water-like fluid to a cluster phase to a gel phase. And what we did was we kind of coarse drained this by looking at patchy. So we kind of coarse drained this entire polymer as a coil, and that could be considered as a soft sphere, which is shown as green here. And this position of this guanidine is actually shown as two red patches. And then by having interactions, ionic interaction between this guanidine, electrostatic interaction between this guanidine, you could actually make a phase diagram where they can either exist as a fluid phase, or they can form a finite size clusters, or continue to grow into a gel, depending upon how much of guanidine is added and what is this strength of attraction, which is modulated by the sodium hydroxide in the solution. So this phase diagram pretty much matched qualitatively with the experimental phase diagram as well. Okay, so moving on, this is again looking at some patchy synthesis that we have done in our lab, again using some sort of interfacial method. The idea is very simple. What we do is we take this charged particle.
3 minutes to go. Okay.
How many?
3.
3 minutes. Okay, then I'll skip this idea because this is something I'll skip. I'll probably go to a project that we have been doing quite for some time now. This is looking at this amyloid B fibrillation. So amyloid beta 40 is one of the very widely studied polypeptide where it forms this kind of a fibrillar network, and it has implication on Alzheimer and Parkinson's diseases as well. So there's a lot of work being done from biology looking at the biochemistry of protein aggregation. So we were actually interested to study this aggregation of protein in the presence of nanoparticles so that we could control the aggregations. So if you look at this, so here we basically monitor the growth of this fibrils using some THT fluorescence assay. So this is basically a pure protein aggregating as a function of time, shows the kinetics. And then we treated this sample with a different amount of gold nanoparticles, and we showed that particles of particular type could actually suppress the amount of fibril formation. We also tried and showed that gold nanorods and silver nanoplates could also do that. But the more important thing is, even though literature has a lot of such studies, we actually found that you could actually have any particle of types, any type of particles, but the charge was the important factor. So the particle has to be negatively charged because the protein that forms this beta sheet has a positive charge locally. And to arrest the folding, what you need to do is you have to have a substrate that has a negative charge so that electrostatically you can bind them and arrest this aggregation. And that we demonstrated with different kind of silica particles, even citrates and CTAP, and then silver particles, gold nanoparticles, and so on. Skip this. And we also built a population balance model to actually study the aggregation of A beta in the presence of nanoparticles using a nucleation, elongation, fragmentation pathways. And as you can see that the experiment and model, the population balance model, could reasonably explain this aggregation kinetics. Okay, I think I'll stop here. Thank you.
Very nice. Lots of possibilities with active particles assemblies and getting the things with it. So I was curious to know that the initial stages of these particles, assemblies and things like that. There's a lot of possibility here in the institute with Chandra here is looking at single particles and several people looking at these. The key point in all of these is let us say, look at your platinum coated polystyrene. There is a chemistry that is there. There is this mechanical properties that are there. There are the bulk issues with surface tension and phenomena of these kind. So I see that this is truly an interface of multiple disciplines. Now, to what extent can one get molecular details of individual chemical reactions, conformation, structures, orientation, directing this overall property, as spanning that entire spectrum? Can one build some models? Can we build some tests, some studies, some experiments, design them, so that I think you can connect between chemistry, biology, and chemical engineering.
Right. Yeah. That's truly, that's right. So in fact, I mean, it's amazing that, how much expertise that we have within our institute. It's just only that what is limiting is that our personal, initiative to, reach out and, for example, like, so far what we've been trying to do is to make collaboration within the group, within chemical engineering or within PEX group. But what I'm really looking forward as part of this initiative is to kind of, step outside my, this comfort zone and talk to people from biology, for instance, because a lot of people working on proteins. I mean, Adi is doing amazing work on protein folding. I'm very fascinated. I sat through his students, compre exams and all that. So it's a lot of things that one can do. So that is from bio. But if you come to chemistry, so chemistry, I mean, you actually in the beginning you actually showed the atomic reactions, particles. You're talking about gold particles, silver particles stabilized by ligands undergoing chemical reactions. That was very fascinating. But what we want to do is as you see, many of the things we are keeping chemistry a little away. We are looking at chemistry to play as an indirect role in changing or modulating the interactions. Because once you get into chemistry, then we are looking at a different time scale, length scale problems. So we want to use chemistry as a way to functionalize the surfaces so that the interaction can be modulated, and then eventually look at the self-assembly of these objects. And as I said before, the reason why I'm saying is because you can just use a simple microscope to visualize these dynamics. Okay, so a lot of opportunities out there. Yeah.
For example, take this particular system that you're having. Instead of one particle now, I connect with the chemical bond these two particles.
Right.
Now they cannot move. So you are probably going to produce a spinning wheel, right? So there are possibilities of this kind that you can build.
Yeah.
With precision. With chemistry, of course, it is precision because it is as precise as a chemical bond.
Right. In fact, one of the
other questions.
Yeah, one of the things we wanted to do was this. As you can see in the slide, that we could actually build. I mean, you can't do this with a spherical particle. Okay, if you have spherical particle, it'll have its own aggregates, irregular aggregates. But by judiciously changing the shape in such a way that you basically allow them to assemble in a particular fashion, in this case, a linear staggered assembly.
Here there is a Venkat, he can connect these two things and get some precise structures.
Okay.
So there are fellows like these who are really good at connecting.
Yeah.
With covalent bonds. I don't know whether he is hearing this.
I am browser.
Venkat, chemistry Venkat, right?
Yeah.
Okay.
So just your neighbor.
Yeah. If you talk louder, I can hear you from here. Just the backside, behind my. Yeah, Venkat, we should probably talk about what we can do.
Yeah. Thank you, bros.
Yeah. Are there other questions?
I have a few. This is Adi here. So, but I think it'll take a bit longer than.
Pick one question and then leave the rest for later coffee table discussion.
Yes. So looks like the potential for there's significant potential for collaboration definitely here. I see. But the one question I had was on the AO model, Asakura Usawa model. So how successful has it been when it comes to predicting the changes in, say, for example, now it's been used to predict a protein DNA and protein-protein binding as well, right? But there is also this effect of solvation that is not really captured by the model. So are there any modifications or variations that one could use, or has it been developed to?
Yeah, so I think this AO model was proposed like in 50s and 60s around that time, and but since then it has gone through a lot of modifications. So basically it's all about depleted, right? So you basically have a larger object, and then you have a soluble small scale solutes, which could be smaller particles, or it could be micelles, or it could be polymer coils. And now then there are a lot of theoretical work that has happened where they look at charge effects. What if the polymer itself is charged? Then whether this AO model will predict the attractive or repulsive interactions is another study people have studied. So there is a lot of development that happened. But as I was saying, people have gone beyond that now. Right now, what is directing this entire field is DNA coded colloids in the last 5-6 years or so. And looks like even that is getting saturated now because so many things have happened. This Shard Mirkin and guys like this, Paul Elvisitos, these guys have done amazing amount of work in DNA coded colloids sort of thing. So
Adi, let us. You will have to take that question with him later.
Yes. I will talk to you.
Krishnarka, you have something quickly to ask?
Sir, I have a two-part question, sir. The first part is like, how you have tracked the motion of these particles like under different pH, and second is like as you can see in the motion, I was able to observe some particles were in focus and some of them were out of focus. So is there any instead of having a 2D spiral, is there a possibility of a 3D spiral motion, and how can we quantify the depth, the bulk motion, sir?
Yeah, so that's a good question. See, these images are optical microscope where you have a fixed focal plane, but there are people who could do actually confocal, so you can do a Z scan. So you can actually scan the entire system in Z scan, and you can reconstruct to see the 3-dimensional motion also. Okay, so that is quite possible with the system. About the first part, the first question that you asked is what? Is it about a pH effect?
The particle tracking, sir. On the slide number 11, where you have seen how the particle has moved, no, sir?
Here.
Yeah. So when
this is again an optical microscopic image. All you need to do is so there is an image J software that our students use. That basically record the spatial position. And
yes, sir. Okay. Okay.
Thank you. Standard things to do in optical microscopy. Yeah.
Thank you. Krishnarka is tracking, particles, aerosols. That is why he was asking.
Oh, I see. Okay. But this is in a liquid. It's in a liquid medium, right?
Yes, sir. I'm trying. My experiments are more on the using compressed air.
Oh, I see. Okay.
Using high-speed camera. So that's difference. Yes, sir.
Yeah.
Well, that is about 100,000, 200,000 frames per second. All right. So today we had 4 speakers, and it was my great surprise that all the 4 people are largely computational people. So in fact, Piyush talked about his experiments in Daji experiments, but then he does computer modeling as well, although he didn't talk about those. So it's something that everybody is getting into, and at probably towards the end of this series, we will have only that left. And so my request to all of you is also pick some experiments in the lab. Of course, you did talk about Ithiraja, you did talk about experiments. So I'm saying more and more of those have to come, and greater number of interactions have to come. And that you rightly said, it is just walking to the neighbor, as we just now saw. This is what it is. Thank you very much. And this will be this is recorded already. And Suraj, you please make sure that this is taken and then stored separately and made available to people when it is done. Thank you very much. Thank you all.
Thank you all. Thank you.